mutation breeding, also known as induced mutation breeding or mutagenesis, is a technique used in plant breeding to introduce genetic variation and create new plant varieties. It involves the deliberate induction of mutation in the DNA of plants, typically through exposure to radiation. These mutations can result in changes in the plant's characteristics such as improved yield, disease resistance, or enhanced nutritional quality. Radiation plant mutation breeding was first proposed by Dr. Louis J. Stadler, an American geneticist in the 1920s. He conducted groundbreaking research on using radiation to induce mutations in plants, particularly through experiments involving X-rays and radium. This marked a significant milestone in the field of plant breeding as it demonstrated the potential for radiation-induced mutagenesis to create novel plant varieties with desirable traits.
Mutation breeding accelerates the process of crop improvement by bypassing the limitation of conventional breeding methods, which often rely on natural or spontaneous mutation occurring at a slower pace. Next, plant mutation breeding can contribute to developing climate resilience crop varieties by introducing traits like drought tolerance, heat tolerance, and resistance to pests and diseases. Plant mutation breeding aids in developing crops with increased resistance to pests and diseases by enhancing the plant's innate defense mechanism, improving its ability to fend off harmful pathogens and reduce crop losses. This also contributes to sustainable agriculture by reducing on chemical inputs such as artificial pesticides. Plant mutation breeding can lead to increased levels of essential vitamins, minerals, antioxidants, and other beneficial compounds in crops, which addresses nutrition deficiencies and improve human health.
Now, let's look at the science behind this, shall we? When high-energy radiation, such as X-rays and gamma rays, passes through living cells, it can directly or indirectly damage the DNA structure. This causes breaks in the DNA strands or altering the nucleotide sequence. These alterations can lead to changes in the genetic code and potentially result in mutations. This damage disrupts the normal replication and repair processes of the cell, causing single-strand breaks or double-strand breaks in the DNA molecule. This leads to replication stalling, error-prone replications, or incomplete replications, which potentially gives rise to new traits and characteristics in the irradiated cells and subsequent generations. But it's important to note that not all mutation attempts has any significant effect on the plant's characteristic. So, how are the desirable traits containing plants successfully mutated?
Now, let's look at the steps. First step is mutagenesis. Plants are exposed to mutagens, which are in this case sources of radiation, such as X-rays, gamma rays, or neutron beams. These mutagens induce changes in the DNA sequence of the plant cells, leading to the occurrence of random mutations. After mutagenesis, the mutated plants are grown and screened to identify desirable traits or characteristics. This selection process involves evaluating the plants for specific traits of interest, such as improved yield, resistance to pests or diseases, altered flowering time, or modified nutritional composition. The selected mutant plants are further evaluated to assess their stability and performance under different environmental conditions. This evaluation includes field trials, laboratory tests, and comparisons with existing varieties to determine their potential usefulness. Once a desirable mutant plant is identified and found to be stable, it can be used as a parent in traditional breeding programs. Crossbreeding with other varieties or cultivars help incorporate the desired traits into commercially viable plant lines. After several rounds of selection and crossbreeding, the new plant variety is thoroughly tested, and if it meets the desired criteria, it can be officially released as a new cultivar.
Now, you might be wondering, how does the study of living things, or biology, is related to crop mutation? Well, biology provides molecular tools and techniques to study DNA, RNA, and protein molecules. For instance, polymerase chain reaction, for short PCR, DNA sequencing, gene expression analysis, and genetic transformation methods enables the characterization and modification of the plant genomes for mutation breeding purposes. Chemistry is employed to analyze and quantify the DNA damage caused by mutagen exposure. For example, techniques like DNA gel electrophoresis, mass spectrometry, and chromatography allow the identification and measurement of chemical modifications or adducts on DNA molecules induced by mutagens.
Physics play a significant role in plant mutation breeding by providing the necessary tools and techniques for inducing by determining the appropriate radiation dosage for plant irradiation. This optimizes the dosage to achieve the desired mutation rate while minimizing harmful effect on plant variability. Mathematics provides essential tools and statistical methods for analyzing and interpreting experimental data such as regression analysis, analysis of variance ANOVA, and multivariate analysis.